It's my pleasure to be hosting the final ACE webinar of this semester. Today, we're talking with James DeGregori about aging and disease, in particular cancer, and if they are inextricably linked, in what ways they're connected, and in what ways they're not exactly the same thing. So really happy that we have James here with us today. So James is at the University of Colorado. He works on evolution in cancer. And James, you have a book that is recently out, which is really about taking an evolutionary approach to cancer, but thinking kind of like very deeply about the environment of the body, right? And the role of that larger environment in cancer progression. So really important set of considerations that come into play when we think about not just the evolutionary process of the cells themselves that might be cancerous, but what's happening in that whole broader environment. So James, it's a real pleasure to have you here, and I'd like to invite you to begin your talk and share your screen, and we will have Q&A. You can ask questions during the webinar if you want, but we'll have about 20 minutes, maybe a little more at the end, to go into more depth on any questions. So we'll look forward to discussing with all of you who are attending remotely as we get to the sort of second half of the webinar today. So with that, James, thank you for being here with us, and please take it away.
Yeah, thanks so much, Athena. Happy to be here and happy to talk about the subject with everyone. So I'll be talking about aging and disease, and must they go together? So I think most people are probably aware that that cancer is a disease of aging, and about 90% of cancers develop after the age of 50. So we can ask ourselves why this is. And so from a conventional perspective, it's thought that this is largely due to the stepwise accumulation of mutations as we get older. But I think we need to challenge that assumption when we start looking at sort of some simple facts. So if we compare different cancers, such as these are different leukemias, AML, CLL, and CML, and you could see that they're age-dependent incidence shown over here. And I'll note that these are known to be initiated by either one or several different mutations in this case. We can also look at solid cancers from colorectal at the top down to mesothelioma down at the bottom. And these, the numbers in parentheses here are the numbers of suspected driver mutations that are required for these cancers. And, they range from, like, for example, from colorectal up to 11, down to mesothelioma, where it's thought to be really one driver. And while those numbers are kind of a bit loose, we don't know exactly what the number of drivers, I think it's safe to say that many different cancers and bodies have very different numbers of drivers, probably varying by at least 10-fold. We also know that all of these different leukemias and cancers initiate in very different stem cell pools. They can be small organized pools like in our gut, where you get on the order of 10 to 20 stem cells per compartment, but tens of millions of those compartments within an adult human gut, to the origin, the cells that from which leukemias originate, which are the hematopoietic stem cells, the blood stem cells in your bone marrow. And those numbers are on the order of about 200,000. So again, orders of magnitudes differences in the numbers of stem cells from which these different cancers arise. And yet, despite these differences in the numbers of stem cells and the huge differences in the number of driver mutations responsible to these cancers, if we do a very simple thing, we simply take all of these different malignancies and normalize them to their maximal incidence, you could see that these patterns fall right on top of each other. So there's a common age dependence of cancer. We also see this for a malignancy or a process known as clonal hematopoiesis that I'll talk about later, whereby as we get old, we get these clones within our bodies that are these circulating cells that are derived from clones of hematopoietic stem cells or progenitor cells that have expanded and that are dominating the blood in a much greater level than one would expect by chance alone. So as I mentioned, there's about 200,000 stem cells that contribute to our blood. So one would expect on average that any given clone on average is going to make up 1/200,000th of your blood. But these make up 10, 15, 20 or greater percent of our blood. And those are also known to be driven by single drivers, and yet they show the same pattern. So I think we need to ask ourselves, what is causing this common pattern of different malignancies in our bodies? And what I'll try to argue is that this is basically, quite simply, just the result of natural selection. And I'm talking about natural selection, over millennia, over many generations throughout our evolutionary history, not just our evolutionary history, but the evolutionary history of all animals. And so the way we can look at this is by considering what have been the selective pressures acting on humans and really all animals, but here we'll focus on humans. And so if you looked at ancient humans, and I'm showing you a curve of Europeans about 15,000 years ago, but very similar curves can be seen for Native Americans 1,000 or 2 years ago within the United States. And they've done similar studies of African populations up to 1,000 years ago in the Ethiopian regions. And the result is very common in that there's rarely survival or much survival past 50. Maybe 5% of the population will live past 50. And so I think what we can appreciate is while modern incidences for cancer and heart disease really start to rise exponentially, starting about 40, but they really get going by the time we're 50, that there is relatively minimal overlap between these curves. So the modern incidence of many aging-associated diseases and the actual fraction of these people alive. And so we could appreciate that there's really minimal selection against cancer and other diseases of aging beyond the age when individuals would already be dead by other causes. And one thing I'll note about these curves is these curves tend to grossly underestimate childhood mortality simply because of what gets preserved in fossil records and so forth. And so you could really think that there's probably a more steep drop right here, followed by this more gradual decline in who's still alive within the population with age. So for these reasons, I would argue life really isn't linear. Our survival is not linear. So this is now showing a curve of survival of humans in the modern world. Modern, actually industrialized worlds like United States or in Europe, for example. And you can see that it's not linear. So for the first part of our life, up to age 40 or so, it's actually quite flat. And in fact, your chance of dying is very minimally different from about your 15th year to almost to your 40th year. So your chance of dying the year before is about the same as your chance of dying the next year. But after about age 36, this starts to change, in which case your chance of dying is greater with every consecutive year than it was for the prior year. I know that's kind of depressing, but that's just the reality. And so this is why we don't just go downhill, we pick up speed. And so while your chance of dying between your 40th and 50th year is appreciable, but not very big, your chance of dying between your 70th and 80th year is really quite large. And thus we lose our probability of survival with each year. And in a modern industrialized society, this is very unlikely to be due to predator. So this is mostly due to essentially intrinsic failings within our body. And even when the cause is extrinsic, like infection, the reason that the elderly died more from COVID is because they were less able to fight it off. So it's really an intrinsic defect, to give one example. And this curve is interesting because if you look at most measures of damage accumulation, DNA damage like mutations, damage to our mitochondria, and other sorts of damage that one can quantify within humans, that damage is largely accumulating linearly across life, at least from maturity. So we have to consider, why is the curve of our decline nonlinear when damage accumulation is relatively linear? And I like to think of this as because of the 3 R's of resiliency. We have a lot of redundancy. I already mentioned we have hundreds of thousands of stem cells. So if you lose some, may not be a big deal right away. We can do replacement. We can replace stem cells. We can replace organelles. We can replace damaged tissue, and we can repair. We can repair the molecules within our body, the DNA, the proteins. We can repair organelles, and we can repair tissues. And so this this investment in our somatic maintenance is critical for allowing this flat period. And you might note, when is this flat period? This flat period is, of course, the very period when we were most likely to be reproductively successful. So this is the direct consequence of evolutionary pressures to maintain us for long enough that we could basically contribute to future generations. And so what are some of these mechanisms to maintain us? And so I won't go into all of these today, but I think these are really important to consider. I think a lot of people will often focus on the cell intrinsic, like DNA repair, maintaining our telomeres, proteostasis, and that's of course important. We have a lot of cell intrinsic quality control. So a cell that gets damaged might commit apoptosis or senescence to eliminate itself from the pool, allowing it to be replaced with a more functional cell. We have the immune system that's continuously circulating, not just detecting foreign stuff, but also damaged stuff within our body that can be eliminated. There's lots of peer pressure, and I think this is often underappreciated, that even cells that get damaged or mutations, they might continue to act and behave normally simply because all the other cells around them are acting and behaving normally. And so there's a certain maintenance of the sort of evolved phenotype because of this peer pressure. And of course, one of my favorites is cell competition, where surrounding cells, the more normal cells, can recognize cells that are either less functional because of mutations or even hyperfunctional, and particularly even malignant cells, because they don't quite conform. And they can be eliminated from the monolayer. They can either be eaten by their peers, which is interesting to consider, or they could be forced out of the monolayer. And in an epithelial layer, if you get forced out of the monolayer, you're basically destined to be shed into the outside world. So this could be in your gut, and you could basically, that cell is going to end up just getting pooped out, or it can be in your skin where it's just going to be shed out into the environment. And so these are mechanisms to maintain functional integrity. And it's essentially a maintenance program for the soma. So why is this important? Well, it's important to maintain the robustness of the organism. So to be able to be fit, and you can't reproduce if you don't survive, if you don't win battles, if you don't avoid predation. And so that's going to be important for reproductive success, is the health of the individual, for your tissue function, for maintaining immune defense, and for avoiding diseases. So maintaining your tissues, as I'll argue throughout, is important for avoiding diseases. What we'll focus on today is on anti-cancer defenses. But I want you to keep this in mind, that everything I talk about, I think, is equally relevant for these other benefits of being, a healthy individual. So in some ways, if we think about this, what I'm really saying is that natural selection has selected in somatic maintenance to ensure or to maximize the probability of an animal's reproductive success. And so in some ways, you could think about our soma, our bodies, is really just a vehicle for the germline, and natural selection has evolved to maximize the efficiency of that vehicle. But there's no point in maintaining that vehicle beyond the ages where it would likely be contributing to the success of the germline. So given this, what does this investment in tissue maintenance during youth and the waning of this investment in old age have to do with cancer? So this is the model that my lab developed, I think, about 20 years ago. Also the title of my book, I'll note, Adaptive Oncogenesis. And it really has two features. And I think in some ways, the first feature may not seem like the more interesting one, but it's the one that's more interesting to me, because it's the part about not changing. And so what we're arguing is that maintaining high stem cell fitness will oppose somatic evolution and thus promote the status quo. And I really should be probably correct this, and it's not just the stem cell fitness, but the tissue fitness, the tissue structure and integrity, because stem cell fitness is dependent on the integrity of their niche, just like it is for any animal in the wild or any species in the wild. So what we're arguing is that if you think about a fitness landscape, and the way we understand fitness landscapes is this Z-axis going to the north is the fitness of in this case, cells, but these could really be any individuals. And the X and Y axis will be all potential phenotypes, which will, of course, depend on the genotype and the epigenotype, the epigenetic profiles of these cells. And so what I'm showing here is a young, healthy stem cell pool and niche. And so in this arrangement, we're arguing that we've evolved stem cells that are well adapted to their niche. And in understanding of evolutionary biology, when a population is well adapted to their niche, they don't tend to change. They're subject to what is known as stabilizing selection, and that change will tend to reduce the fitness of the individual. And we argue the same thing for these stem cells. The change will tend to reduce the fitness, and that's even if the change has a cancer-associated mutation, an oncogenic mutation. That will reduce the change because the cells were already well adapted to their niche. That's the evolved state. And given that, even if there was a malignant genotype and phenotype that could have a higher fitness, which we think is quite probable, on the same landscape, if getting there means going through low fitness intermediates, that transition will be improbable. And I'm using words like improbable on purpose and not impossible, because a 30-year-old who does everything right, eats a good diet, exercises, can still get cancer. Just her risk of getting that cancer is much, much lower, often logs lower than would be the risk of a 65-year-old who smoked, for example. So natural selection is not seeking perfection. It's not creating organisms that will never get cancer till they're old, but it reduces risk to the organism to the extent possible until they are beyond the age of likely reproduction. Of course, this is all nice. This can explain why we don't get so much cancer when we're young, but why does this end up changing? Well, it ends up changing because of the reasons I already told you, is that we haven't had selective pressure throughout our evolutionary history to maintain us beyond the ages where we would likely be contributing to future generations. So given that, our tissues go downhill. We can also imagine that we do things that we shouldn't do. We smoke cigarettes, for example. So let's assume that these are now stem cells in lungs. So in a young, healthy person's lung, that stem cell may be well adapted to its environment. But in a smoker's lung, or in the lungs of an older person, that relationship may be changed. And that stem cell might find itself in a niche that it's no longer well adapted to. And that state of poor adaptation is going to now select for variants that are better adapted. And that could lead up a path towards malignancy. What I'm not showing here, but I think you'll hopefully appreciate from what I'll be telling you in a little bit, is that this isn't limited to malignant evolution, that when you get changes in the microenvironment, you can stimulate a lot of non-malignant evolution as well. And there's probably many new paths created by the state of poor adaptation that could lead to clonal evolution within our tissues, only some of which might lead to a cancer. So then what is this evidence for differential selection for somatic mutations depending on context? So a very talented postdoc who was in my lab a few years ago, Curtis Henry, who's now faculty at Emory University, wanted to test this. And he tested this by doing a very simple thing. He took early stem cells from mice, introduced an oncogene into these stem cells, and tagged them with the GFP, which is a green fluorescent protein. So this oncogene is RAS. And then he put these into mice. So all of the cells he's putting in are young, but he wanted to ask, if he put these into mice at different ages, would he definitely get different consequences from this introduction? So these mice were mildly treated so that we weren't disrupting the environment, and they're either young or they're old, and he blocked inflammation as a way of sort of altering this aging associated microenvironment. And when he did this, the result was quite dramatic. First off, quite surprisingly, the RAS expressing cells, despite expressing an oncogene, did not win in the environment of the young. In other words, they were not well adapted in the young environment. Sort of suggesting that, again, as I mentioned before, the stem cells that are already in the young environment are well adapted to that environment. But in the old environment, those same cells no difference in the cell with the oncogene, but now these cells are in a background of an aged individual, and now they expand out. So this oncogenic event was adaptive in the aged environment, but not the young environment. But importantly, when he blocked this aging-associated inflammation with this IL-37, he prevented that selection. So it also says that one can change the selective advantage of an oncogenic event by manipulating the environment. You don't actually have to target the cells themselves. He went on to show why this is, and he looked at signaling components, and he showed that these signaling components are down in the old, but they're restored by the activated RAS oncogene. And this can explain why it's adaptive in the old environment, because things aren't doing well, so there's something to improve upon, but not the young environment, where it's already got the optimal amount of RAS signaling. And it also explains why this IL-37 prevented this adaptation, because it prevented much of that drop in the first place, thus taking away the selective advantage of having RAS. And I won't go into the details, but he sees the same things when he looks at gene expression: a drop in old, restoration by RAS, and a partial or full prevention of that drop in the first place by expressing this IL-37 transgene. So now I'm going to turn briefly to clonal hematopoiesis, which I referred to before. So as we get older, and typically this starts after the age of 50, as you know, normally as we age, our hematopoietic stem cells are accumulating mutations. Just because they undergo division, just because they're in our body, time and mutations go hand in hand. But most of these mutations will be what we would call passenger mutations. They're relatively inconsequential for the cell, and they don't affect its ability to compete with other cells within the hematopoietic stem cell pools. They're almost just markers of time. But you can get these mutations that can increase the fitness of the cell and can lead to clonal expansion, so allowing them to outcompete their peers. And so, as I mentioned before, despite this stem cell being, let's say, 1 in 200,000 stem cells, this stem cell can then give rise to a clone that now occupies 10, 20, 30% of the blood of this individual. And this will happen in about 20% of the elderly people. And it's thought to be a precursor for blood cancers. In other words, we know that you could look at some of these people over time, and you could, let's say you have someone that comes down with AML and you happen to have a blood draw from them from 10 years earlier, you might find the initiating clone of their AML in their blood 20 years earlier. And so you know that that was the precursor to their AML. But what's, I think, relevant is this clonal hematopoiesis is not just associated with more leukemia. It's associated with more cancers in general. It's associated with coronary heart disease, all other sorts of cardiac failures. It's associated with lung problems, and it's even associated with overall mortality. So these clonal expansions probably are doing much more than just contributing precursors for leukemia. In fact, it's the clone itself and its abnormal features. In fact, it's the features of many of the mature cells that are produced from this clone that are probably contributing some of these other aging associated problems that we encounter. And again, this is why I want everyone to be thinking about not just cancer, but aging associated diseases in general, and that they can often have common causes. So we wanted to study this, and we picked one of the two most common mutations observed in this clonal hematopoiesis, TET2. And TET2 has also been directly implicated in contributing to coronary heart disease through its contributions to hematopoietic stem and progenitor cells. And so we being Marco in my lab, shown over here in the right, a postdoc in the lab, developed really elegant methods to basically knock out any gene he wanted in hematopoietic stem cells in a mouse under relatively non-perturbed context. And he's doing this either in young or old mice. So he takes bone marrow, isolates their stem and progenitor cells, and then he uses CRISPR. And I'm sorry, I won't have time to go into CRISPR, but hopefully people have heard of it. It's his method where you can very selectively knock out any gene you want. And that's really all you need to know. And he introduces that in along with the control that makes basically a mutation somewhere else in the genome that shouldn't matter. and he puts these into the cells, and then he can put these back into the recipients, which are either young or old. And just suffice it to say that this method's quite efficient. And what he found is that the age of the host mattered. And so when he goes into the young host, he gets a certain amount of the contribution of these cells to the peripheral blood, and he gets more of that in the old. And he gets that across all these different lineages of cells that he's looked at. These are T cells, B cells, these are macrophages and granulocytes. In all of the cases, he's getting more in the old than he does in the young. And importantly, when he looks in the bone marrow, specifically at stem and progenitor cells, he sees the same thing. So these are, for example, early committed progenitors, more in the old than the young. And these will be enriched for more hematopoietic stem type cells. And again, more in the old and the young. And this is specifically looking at hematopoietic stem cells, and you get more in the old than the young. So the effect of this mutation is not the same in a young individual as it is in an old individual. The young individual, being a mouse in this case, is better able to restrain that mutation and prevent it from expanding. So the reason we have more TET2 mutant clones in the elderly people than in young people isn't just because of time, it's because the actual mutation has a differential effect on the fitness of the cells depending on the age of the individual, with a greater effect in old. So what are the conclusions? We can conclude that old age promotes selection for TET2 mutation. This would also indicate that we might be able to develop interventions that can reverse this aging-associated increased selection for TET2 loss. And we could use these mouse models as a means to do that. And we can ask then, would such an intervention not only reduce the resulting increased leukemia risk that would be initiated by a TET2 mutation, but also other consequences of TET2 mutant blood cells, such as coronary vascular disease. So another thing I'd like to touch upon is that as we get old, it's not just our hematopoietic system that becomes dominated by these different mutations, these different mutant clones. It's our entire tissues, and a bunch of different studies, and I won't have time to go into them, have shown that our tissues basically become, basically filled with these different clones, often driven by cancer-associated mutations as we get old. And so another postdoc in my lab, Dr. Evans, decided to sort of quantify this and better sort of analyze all these different mutations that have been seen in all these different studies. And so he took all of these different studies and pulled the different sort of the data from all these studies and then passed it through similar filters. So for example, in some cases, he would ask whether the mutation was present in the cancer gene consensus, or the CGC. So this is basically, has the mutation been seen in cancer, and is there reasonable odds that that mutation is contributing to cancer? He also asked whether the mutation was what we call pathogenic. And this simply means that it's disrupting protein function, or it's predicted to. And what you could see is with age, you get this increase, and look at the ages groups are shown down here on the bottom right. You get an increase in the contributions of these clones to tissues. But I'll note that this varies from tissue to tissue. So for example, in the bladder, it's typically 20% or less. While in the esophagus or the colon, it can reach close to 100% of the tissue can be occupied by cells with known driver mutations. It's kind of scary, except that we also know that only a few percent of us will get colon cancer. So apparently for most of us, this isn't going to cause a cancer. But I think that's quite striking. And when he quantifies this, by the time you're 60, you have over 100 billion cells that harbor at least one oncogenic mutation. This is out of a few trillion nucleated cells that you have in your body. So it's a massive percent of your body is occupied by cells with known cancer drivers. And yet only about 40% of us will develop cancer, which will almost always arise from a single oncogenically initiated cell. So what this raises is, what do the other 100 billion or so cells with oncogenic mutations do? And that's what we'll talk a little bit about here. So just to summarize what I've been telling you so far is that we get this accumulation of genetic damage throughout our lives. but at least through youth, we feel that this is suppressed to some extent. And this suppression is not only pushing cancer off to the right side of the curve, but it's also probably preventing a lot of the other consequences of these clones to our normal tissue function. And I'll get more into that. But late in life, as the maintenance of our tissue has waned for the evolutionary reasons that I went into, and now our tissue structure and function is going downhill, that this allows opportunities for positive selection, for adaptation that could then contribute to a cancer as we get older. So if you think about this, when we think about pathogenic clonal evolution, like these TET2 mutant clones, one thing that's already been shown is that these mutant clones themselves can be inflammatory. But this inflammation itself can then contribute to selection for the pathogenic mutant clone. And these clones can contribute to cancers and to leukemias, as we've discussed. But we also need to be thinking about how these different clones could contribute to other diseases of aging that we should care about as we get older. So I'm going to quickly turn to a last example. And I'm actually, I think I've got about 10 minutes left if my timing's about right. To talk about how smoking status will influence oncogene-driven clonal expansion in the lung. So I've been talking about aging. And of course, that's kind of depressing because there's not a whole lot we can do about that. But here's something you can do something about. You can not smoke. And it's understanding how an extrinsic factor like smoking can influence somatic evolution within our bodies. And so in this study, we were able to get biopsies from the contralateral lung, and that's a normal lung. It's contralateral to either a detected tumor, but these are by CT scans. So, and about half the times there was no tumor. It was actually a false call. But either way, we've got these contralateral biopsies that either were across from the lung where the tumor was or where there was not a tumor. And we're using this method called duplex sequencing. And I won't go into details, but essentially this allows us to detect mutations in a very error-corrected manner because we separately sequence the two strands of DNA. And so the only way we end up believing a mutation, if it's present in all of the reads that we get off the sequencer, and also that it's present in both strands. And the chance that that would happen, by chance alone is less than 1 in 10 to the minus 7. So less than 1 in a million, 1 in 10 million, I should say. And so we can very confidently call these mutations as being real. And when we look at these biopsies from people, and this is about a 2 centimeter squared brushing from the epithelium of these individuals, we can see that we get dozens, if not hundreds of mutations per sample. They're all piled up on top of each other here. And these mutations are varying in what we call their VAF. Their VAF is the variant allele frequency. And what you should know is if you took the log of this, so a -3 VAF means that about 1 in 1,000 cells have this mutation. And a -1 VAF will mean that 1 in 10 cells has this mutation. And so the further to the north you are on this curve, the bigger the clone. It means that the much bigger clone has this mutation than if you're down here at the bottom. And if you look at the involved site, you can see that we do see, particularly in those that have cancer, some very high variant allele mutations. In fact, these are known cancer drivers that were characterizing the cancers of these people. And from now on, I'll only be talking about the contralateral side. So I'm only going to be talking about tissue that does not come near the cancer. So now if we just focus on the contralateral side and we separate out people based on their smoking status, never, former, or current, we could start to then look at the cancer-associated mutations that are found. So what I'm showing you now is now calling everything I showed you before, but only for cancer-associated and damaging mutations. And what you note is that, or first off, I'll note that the ones in yellow is where individual had a lung cancer in the other lobe. And what you'll basically notice is that it doesn't really matter. I mean, basically normal seems to be normal, independent of whether there's cancer across from it. But if we do notice is that the never smokers have very few mutations that go to very high VAFs, meaning where they're big clones. We see more in the former and current smokers. So essentially smoking is influencing selection for the oncogenic variants. And this is just showing you some of these oncogenic variants, and it's really dominated by Notch and P53, which are the blues and the pinks. We see the same thing for indels, which is small insertions and deletions, and it's really dominated by Notch mutations. And this is just showing you significance that the never smokers have significantly fewer mutations, are smaller clones of these mutations than the people that smoked. So just to show you an example, this is PIK3CA. We only see it in the former and current smokers, and these are actually the mutations that are directly found in cancers, which is what's shown on the x-axis here. This is the frequency that these are found in human cancers. Same thing for PD3, is we're really getting some of the most common mutations found in cancers. And one thing I'll note is that the top 6 mutations were all from the current smokers, suggesting that perhaps as one quits smoking, the size of these clones will start going down. So then how are we going to go about looking further at these different mutations? We're going to try to understand not just how they might contribute to future cancers, but how they might affect normal function. And we're going to be using culture systems. These air-liquid interface cultures basically allow us to create a stratified epithelium in culture, and then ask, how does introduction, for example, of a Notch1 mutation affect the function of the lung? We'll do the same in mouse lungs. And we can ask, what's the impact on function, and as well as oncogenesis, with or without smoking, for example. So now I'm going to very briefly turn to this study that we're doing with our collaborators in England, the group of Charlie Swanton. And
James,
for this,
do we have a clarification question about the smoking? If there was
Sure.
an average or minimum number of years for former smokers?
Yeah, and well, they're almost always for decades, but that's always the problem when you do these clinical studies, because basically we can't control it. This is anyone who comes in for a CT scan. So usually the CT scan is mandated for people that are over 65 and that smoke for a certain number of years. And I forget exactly what that is. I think it's at least 15 pack years, which is basically if you smoke 1 pack a day for 15 years, whether you're current or former. But yeah, and unfortunately, given that we are only, we're limited in what we can take, it's not exactly uniform.
Okay.
So it's really all comers. But great question. Oh, so the piece is what I'll be talking about, the piece samples, and these are from an autopsy program. And so we're essentially going to be looking at histologically normal lung biopsies from individuals who had cancer elsewhere. And what you could see is now we have many more never smokers, because unlike for CT scans, where it's rare to have never smokers come in for a CT scan, we all die even if we're never smokers. So you can get never smoker samples, and you can see that we're getting more mutations that pass our criteria in the ever smokers than the never smokers. And for ever smokers, we're combining former and current smokers. And the same thing when we only ask for mutations that are present within the cancer gene consensus, which is known as cosmic. We get less of them in the never smokers. And this is less per sample is what's being shown here. But more importantly, from my perspective, because it's a direct measure of selection, is the size of the clone, which is measured by VAF. So we get bigger clones in the people that smoked relative to the people that never smoked. And this is for only looking at KRAS, and we get the same thing when we look at EGFR. And so now for the very final thing, I'm going to show you something that I think is really exciting, is because one thing we were able to get from these autopsies is you can get the whole lung. And we could take that lung and do biopsies across the lung, we being the people at the CRIC, they sent us the samples. And so we essentially have this lung with all these different small little biopsies taken out of it. We then did our duplex sequencing on the lung samples. And what we get is really striking, is that we see different oncogenic variants throughout the lung, but even in the same gene. So for example, you can see over here for BRAF that we get different BRAF mutations, all of which are known to be cancer associated, but you get different ones at different regions of the lung for these different people's lungs. And I'll note that all 3 of these people are old. The first 2 are never smokers. The last one is technically a former smoker, but this person quit 50 years ago. So in some respects, I think we can consider all pretty close to never smoker. And this is just focusing on one gene. And I just want to point out the differences so you can see these different oncogenic variants in different regions. So it sort of suggests that this landscape of the lung is highly selective for PIK3CA mutations for whatever reason. And it'll find different ways of doing it. So it's like convergent evolution towards the same end, but at different points within the lung. And you can see that this other person seems to have less pressure for getting the same outcome. And we could see this more clearly when we look across, everybody, and you can see that there are differences in how selective these mutations are in different individuals. And for the last data slide, I'll show you this in a really geeky way, which is looking at direct measure of the power of positive selection. And I won't go into what DNDS is, but anything over 1 is an indicator of positive selection. And you could see that for many of these genes, we're seeing a lot of these regions, each dot is a region that show clear positive selection for that gene within that individual. But I'll note that some of these individuals, most of the regions are not showing signs of positive selection. And for others, almost every part of the region, every part of the lung is showing such positive selection. I'll note that there are control regions where you get absolutely no change at all. In fact, we don't even detect mutations in it. So this is not random. There are certain selective pressures happening within these lungs. So what this tells us that smoking promotes selection for oncogenic variants in the lung. These clones are predicted to impact lung function and contribute to cancer risk. And we really can ask, can we develop interventions that accelerate this restoration of more normal lung landscape after quitting? And what this would look like is you can imagine that when you quit smoking, with each subsequent year, your risk of cancer goes down. That's the good news. But the goal of an intervention would be if somebody could take something or have something that would accelerate and deepen this reduction in lung cancer risk. And what we're arguing is that this mutation detection method and looking at these different clones and their sizes could be a proxy for that. That this may be a proxy for this risk that would allow us to more rapidly develop these interventions without having to wait till the subjects developed or did not develop cancer. I'm going to skip this one for time and just go to this last one. So what I've been trying to tell you is that aging, we know through mutations, through altering adaptive landscapes, altering immune function, and inflammation can cause clonal evolution in our tissues. And that can, of course, lead to cancers. But what I want you to appreciate is that this clonal evolution itself, by promoting inflammation, perhaps by reducing barrier function within our epithelial tissues, or by reducing tissue dysfunction, could actually be contributing to aging. So we can't think of this as just a one-way street anymore. So with that, I think I've thanked people and the collaborators along the way, and of course, my wonderful lab. And of course, all the wonderful funding we get. And here's an actual picture of my book. And I'll note that all that wonderful artwork you saw was from my son, Michael, and he did the artwork for my book. And many of those pictures were stolen from my book. So with that, I'm happy to take questions.
Thank you so much, James.
Should I stop sharing? Would that be the best thing?
Yeah, why don't you stop sharing? And then if people do have specific questions about slides, you could always go back. But yeah, it's great to know that you stole the images from your own book. So I don't think it's stealing, actually, if you just use the images from your own book. So I want to invite everybody to put questions into the webinar chat so we can do some Q&A. And while you get your thoughts together for that, James, I'll start in with a sort of question or maybe sort of more of a sort of broad discussion point. So, this general framework that you're putting forward of, really thinking about sort of what's going on with the environment that the cancer cells are evolving in, and how does that environment change over time? There are a couple of really interesting ways to sort of think about that if you kind of layer, cooperation theory on top of this too, right? Mm-hmm. So if we think of multicellular body as this cooperative entity, and it evolved to, limit cheating, promote cooperation. So one way that we could think about what's happening with aging is that, yes, we have all these regulatory systems. We have all of these, genes that give us the ability to cooperate on a cellular level and monitor cheating on a cellular level. And that, as we age, those systems are less fresh in a way, right?
Yeah.
And so there might be a sort of breakdown of the sort of fundamental cooperation, breakdown of the sort of cheater detection systems that's happening as a result of aging. So that's one possibility, right? But then another possibility is that a lot of what is going on with aging is that the trade-offs for actively trying to suppress cancer are increasing. And, the side effects of having a lot of, responsiveness, changing your expression state, activating the immune system, all of those things might be going up, leading to sort of a functional, down regulation of some of those systems. So I'm wondering, on what side of that do you lean, or do you think that both are going on and interacting with each other as we age and our ability to suppress cancer changes?
Yeah, definitely. I think both are at play. I memean, for the first one, I think it could be sort of, a probability type issue, and a feed forward loop versus a feed forward loops that could happen both to our benefit and to our detriment. So as you talked about, like there's ways of getting rid of cheaters, and in some ways it's not just getting rid of cheaters, but it's getting rid of poor workers too. So if you imagine a population of epithelial cells, the surveillance systems are not just for a cell that might be misbehaving to become more malignant, but also a cell that might be misbehaving and that's just not functioning well. So, and some of these are even, directly sort of instituted by damage type systems. So there's a beautiful paper that shows that if you get UV damage to stem cells within our skin, that cell will no longer attach well to the basement membrane, and it loses some of those attachment points. And at that point, its neighbors are able to physically force it up. Now that cell doesn't become useless because it's just going to go above the basal layer, but without that contact to the substrate, it's no longer really a stem cell. And now it's going to make contributions to your skin. But as we know, our skin turns over at a high rate. So essentially it's making contributions, but they're now temporary contributions. So in some ways it's a better safe than sorry. It's not that the system knows that cell received damage that would never necessarily cause it to be oncogenic or even cause it necessarily to be poorly functional. But the system is like, well, we've got lots of skin cells. Let's just get rid of that one. So those sorts of systems, though, are completely dependent on the surrounding cells being healthy and so forth. And so if the surrounding cells were less healthy, their ability to eliminate that peer starts going down. So let's say that I'm just totally guessing these numbers, but let's say the efficiency of eliminating is 97%. And so that's what happens in youth. But with time, you can imagine that you're going to now have some of these cells that didn't properly get eliminated because they were among the 3% that didn't. And now there's some chance that you're going to get a cell that sits down. It's got damage. It needs to be eliminated, whether that damage is going to be oncogenic or not. And it happens to be next to another cell that didn't get eliminated before. And now its chance of getting eliminated is 85% because it's got some of its neighbors that aren't so great anymore. But with time, you can imagine that's going to be a logarithmic increase in the odds that something doesn't get properly eliminated as more and more of the other cells are essentially less functional to begin with. And so that would be a feed forward loop that in youth, the feed forward is the more healthy the tissue, the better you are at eliminating. But as you move forward, it's the less healthy the tissues, the less you are good at eliminating. And now the more likely that you're going to get even worse at eliminating. So that would be sort of the cheater system is going to go downhill because the cheater system is dependent on having lots of healthy cells. That's on the one side. And I think in terms of this sort of antagonistic pleiotropy, which is like what's good for us in youth, maybe isn't so good for us in old age. I don't know if it's quite that simple. Take inflammation, for example. Inflammation is still good for us in old age, but we know that being hyper-inflammatory in old age can be a problem. But if we would just simply say, well, then let's just suppress inflammation in old age, well, then people die of infections. So it's not that inflammation is unimportant, but we also know that inflammation is a contributor to our decline in old age, a contributor to cancer risk, a contributor to heart disease. So what we need to learn is we need to figure out how to tune, because what we'd really like is to have the inflammation of a 30-year-old and not the inflammation of a 70-year-old. That's not easy, but that's sort of the antagonistic pleiotropy. Those very same things that were so important for maintaining us our youth are now coming back and biting us in the rear when we're old. But it's not so simple that we can just say, oh, now we can just get rid of them.
Yeah. Well, I would like to invite the audience again. Please, please give us your questions and your messages, your comments, so we can have a broader discussion here. In the meantime, James, it looks like there was a question maybe asking about the signaling that was going on in the old subjects, old participants that were RES positive.
Right.
Yeah, so maybe we can, if you don't mind, maybe sharing your screen again and going back
Sure.
to that and explaining what was happening with the signaling. So I think, right, that was basically a
that was in the mouse
situation,
model, yeah.
yeah, where there was a restoration of some function. Is that right?
So this is here. So basically, these are cells from young mice. So these cells that are being shown here are actually the young progenitor cells for the blood. And these cells, what we're looking at here is a measure of the phospho-STAT 5 signaling. So it's a measure, it's a critical signaling component for that is key to the fitness of these cells. That's known from other studies. And we show it goes down in old age in the old cells. So even though these are young cells, they're in an old mouse, and so they don't signal as well. And it's a point I probably should have emphasized more, but if they have RAS activation, they can now have a more normal level of signaling. And so essentially, and so you can see why if you put RAS into these old cells, if you can imagine this being sort of a proxy for fitness, well, then you can see why these RAS clones are going to be more fit than the wild type, causing them to be clonally expanded. On the other hand, you could see why, if you can imagine that this is the evolved amount of phospho-STAT 5 that one would have, this would be the right amount, that more of it's not going to be better. And so having RAS in the young, you can see why that was not selected in the young, because they already had the right amount of RAS signaling. Does that make more sense? And then finally, when we put in the IL-37 transgene into the old, it prevents that reduction from happening. Instead, they're maintenance. At least it seems to be good enough to prevent that selection from happening. And we're looking at this with other parameters over here. These are involved in nucleotide synthesis and cell cycle progression, but it's always the same thing. These parameters are always reduced in old, restored by RAS expression, and at least to some extent restored in the mice that have the IL-37 transgene.
Great. James, we have a kind of a big picture question, which I think dovetails a little bit with some of these topics, which is, if you sort of look at these oncogenic mutations that are occurring in normal cells during aging, are those mutations contributing in a positive way to extending tissue function, or is it always problematic?
No, in fact, I think it's a great question, and it's probably complicated. And like I mentioned, I mean, we're taking 2 centimeters squared of somebody's lung. So the chance that any of those mutations, where we're seeing hundreds of them, actually went on to contribute to cancer is infinitesimal, right? Because even of the people smokers that we took, only 1 in 7 of those smokers can get cancer, and they could get it anywhere in the lungs, and it could be any from those cells. So we really need to understand, what are these cells doing that's not contributing to cancer, which is not normally how cancer biologists think. But there is a very clear example of mutations that do clonally expand in our tissues as we get older that seem to be, if anything, preventing of cancers. And they're these Notch1 mutations. And they've been pretty clearly shown in the esophagus. So in the esophagus, if you're over 50, the majority of your esophagus is constituted by these clones with Notch1 mutations. You have more esophagus that has Notch1 mutations than you have cells that don't have them, which is pretty amazing to think of. This is even in never smokers, people don't drink heavy. In other words, you've done everything right, and your esophagus is getting constituted by this. And yet, when we look at cancers from the esophagus, they rarely have Notch mutations, about 10, 15%. So in other words, it's, if you just thought about it by chance alone, let's say Notch mutations didn't do anything to the cells whatsoever, you would expect over half of the cancers to have Notch mutations, because over half the cells have Notch mutations, but that's not the case. So suggesting that cells with Notch mutations are actually at an advantage for not becoming a cancer. They're selectively not becoming cancers. And so we've actually proposed that this is what we call a decoy fitness peak. So that I mentioned before, when you look at these adaptive landscapes, you can get adaptation up a malignant peak. Well, this would be adaptation up another peak, but it's essentially a more benign alternative to having gone up that malignant peak. And so we think this can happen within our tissues, but that's not to say that those Notch mutant cells are equally functional as a wild type cell. It may be a bit of a trade-off in the sense that, well, maybe they're not quite as functional, but hey, at least they're not malignant, right? So I think that's about as, hopefully that makes sense to the individual.
Yeah, great. Do you see that as I'm sort of taking up the ecological space so that it's not available for cancer cells?
Exactly. It's essentially, taking, it's now spreading out a clone. And so you can imagine that, as these clones dominate our esophagus, those are basically now that option for malignancy is no longer available, because they're already up a new peak, and going down and back up a malignant peak would be difficult.
Yeah, great. We have one final question here from Nadia Wright. Nadia is asking, so within your lab, have you looked at or thought about interventions applied to other carcinogenic exposures, so other than smoking?
No, we're looking at, certainly in the context of aging, we are looking in the context of smoking. One of the reasons we focused on smoking is because if you ever want to make an impact with this stuff, as much as I'm interested in aging in general, it's going to be really hard to institute something that would be so population wide. It would have to have essentially zero negative consequences, and it would have to be cheap. So there's a lot of difficulties with that. But smokers represent a population that's obviously only a fraction of the population. They already know they have a high risk for cancer. And they're already coming in anyway, for example, based on recommendations from the government, for example, to get these CT scans. So we're kind of capitalizing on that and to develop interventions. So we have a lot of studies with that, everything from dietary change to interventions that are promoting autophagy, which is a tissue maintenance function, to anti-inflammatories. Although with the anti-inflammatories, I really worry that to actually institute those is going to be very difficult. But with aging, we're looking at some that's simply aging based. We haven't been looking at other sort of inducers of cancer quite yet beyond aging and smoking. That's really what we've been focused on.
Great. Thank you so much, James. Do you have any final thoughts for us, or anything that you'd like to leave us with before we say goodbye?
Besides don't smoke, let me see. I think that if we think about, how our bodies change as we age, I just think we really need to look at things more holistically and not become, at the same time that, I think we all have to be reductionists sometimes, because if you don't become reductionist, you can't understand something mechanistic. But I think you have to incorporate it into a larger framework. And that not only means the framework of the whole body, a holistic way of looking at things in that way, but I think we need to think about it in an evolutionary framework. If it doesn't make sense from an evolutionary perspective, and I'm talking about, over, geological time, then there's probably something wrong with the way you're thinking about it.
Great. Well, thank you, James. I think that's great sort of general thoughts to leave us with, that, taking an evolutionary approach to looking at not just cancer, but health and disease more generally can kind of keep us on the right track, right? And
Exactly.
and thinking about these evolutionary pressures, not just on organisms, but also within organisms. And that might be relevant not just for cancer, but for thinking about disease much more generally and what our vulnerabilities are. So
Exactly.
yeah, so thank you so much for sharing your work with us and having this discussion with us as well. And thank you to all of our webinar participants. Thank you for your questions. Thank you for your attention. And we look forward to seeing you again in the next semester for our ACE webinars in the future. So until then, don't smoke and think about evolution on multiple levels.
Great. Thanks so much and have a good rest of your day.
Thank you. Bye-bye.